Hello students, I am Ashok Waghe from Fergusson Junior College. So here I am in front of you once again to continue the lesson that is called as molecular basis of inheritance. During the previous lecture we completed about the protein synthesis, that is how the transcription mechanism, how the translation mechanism it is there. Hence, in this lesson, we want to continue that part only, that how the gene it is controlled, or the expression of gene, how it is controlled. So it can be controlled at the transcriptional level or translational level. In addition to that, the second part that about the genome and genomics we want to complete. And the last point we want to complete, that is called as the DNA fingerprinting. So that's why let's start with that of the number one point, today's number one point, that is called as the gene expression and its regulation. Okay, so let's start with that of the gene expression and gene regulation. Regulation of gene expression with Lac Operon model in front of you. The Lac Operon model also I kept in front of you, the diagrammatic representation where the regulator gene, the promoter gene, the operator gene, and the structural genes. So let's see about the introductory part, then I will explain how the regulation of gene it is there. It is the multi-step process by which a gene is regulated and its product is synthesized. Thus, gene expression results in the formation of a polypeptide. Gene expression process is regulated at different levels. In eukaryotes, the regulation can be at different levels like transcriptional level, that is formation of primary transcript, processing level, that is regulation of splicing, 3. Transport of mRNA from nucleus to the cytoplasm, and 4. Translational level. Genes of a cell are expressed to perform different functions. For example, an enzyme beta-galactosidase is synthesized by E. coli. It is used for hydrolysis of lactose into galactose and glucose. So the chemical reaction it is like this, that lactose in presence of beta-galactosidase and in addition to that water. So galactose and glucose formation it is there. If E. coli bacteria do not have lactose in the surrounding medium as a source of energy, then enzyme beta-galactosidase is not synthesized. So it is the metabolic or physiological or environmental conditions that regulate expression of genes. The development and differentiation of embryo into an adult organism is also a result of the coordinated regulation or expression of several sets of genes. Now one has to understand and know the mechanism by which the organism regulates gene expression in response to changes in the environment. If so, whether single mechanism exists for regulation of the expression of different genes or sets of genes or different genes are regulated by different mechanisms. Certain bacteria like E. coli adapt to their chemical environment by synthesizing certain enzymes depending upon the substrate present. Such an adaptive enzyme is called inducible enzymes. A set of genes will be switched on when there is necessity to metabolize a new substrate. This phenomenon is called induction, and small molecule responsible for this is known as inducer. It is positive control. So Lac Operon model. So let's see its explanation. Operon concept. Operon concept is a transcriptional control mechanism of gene regulation. Francois Jacob and Jacques Monod in 1961 explained that metabolic pathways are regulated as a unit. For example, E. coli, when lactose sugar is provided to the culture medium, cell induces production of 3 enzymes necessary for digestion of lactose. The enzymes are number 1, beta-galactosidase, digests lactose into galactose and glucose. Beta-galactoside permeates lactose molecules to enter into the cell. Trans-acetylase, beta-galactoside trans-acetyltransferase. This is also called as the beta-galactoside acetyltransferase. Transfers an acetyl group from acetyl coenzyme A to galactoside. Synthesis of these three enzymes is controlled by a long segment of DNA known as operon. It consists of, see in the diagram properly, it consists of an operator site O and three structural genes Z, Y and A. The action of structural genes is regulated by operator site with the help of a repressor protein. A repressor protein is produced by the action of gene I, that is inhibitor, known as regulator gene. The gene expression depends on whether operator is switched on or switched off. If the operator is switched on, the three genes Z, Y and A are transcribed by RNA polymerase into a single mRNA. Each structural gene is generally known as cistron, and the transcribed long mRNA covering various cistron is known as polycistronic. Switching on or switching off of the operator is achieved by a protein called repressor. When this protein is attached to operator and blocks it, the switch is turned off and structural genes are not expressed. Lac Operon. Lactose or Lac Operon of E. coli is inducible operon. The operon is switched on when a chemical inducer lactose is present in the medium. Thus, Lac Operon can be explained as follows. The Lac Operon consists of regulator gene, promoter gene, operator gene, structural genes and inducer. See, I kept in front of you everything with the help of these two diagrams. So whatever I am explaining, you please refer the diagram which is there in front of your screen. So number one, I want to explain here, that is about the regulator gene. This gene controls the operator gene in cooperation with an inducer present in the cytoplasm. Regulator gene precedes the promoter gene, that it is found before the promoter gene. It may not be present immediately adjacent to operator gene. Regulator gene produces a protein called repressor protein. See the diagram number 1, repressor protein. Repressor binds with the operator gene and represses or stops its action. It is called regulator protein. Number 2, promoter gene. This gene precedes the operator gene. It is present adjacent to operator gene. The promoter gene marks the site at which the RNA polymerase enzyme binds. When the operator gene is turned on, the enzyme moves over the operator gene and transcription of structural genes starts. Promoter gene base sequence determines which strand of DNA act as a template. Operator gene. It precedes the structural genes. This controls the functioning of structural genes. It lies adjacent to the structural genes. When operator gene is turned on by an inducer, the structural genes produce mRNA. Operator gene is turned off by a product of repressor gene. Structural gene. When lactose is added to the E. coli culture, so I shown the culture medium also in the diagram, please refer that. The structural genes catalyze mRNA, which in turn produces polypeptides on the ribosomes. The polypeptides act as enzymes to catalyze lactose in the cell. There are 3 structural genes in the sequence: Lac Operon Z, Lac Y, Lac A. Enzymes produced are beta-galactosidase, that is Lac Z produce beta-galactosidase, Lac Y produce beta-galactoside permease, and Lac A produce trans-acetylase respectively. Inducer. It is a chemical in the cytoplasm which inactivates the repressor when Lac Operon is switched on. Then inducer joins with the repressor protein, preventing the binding of repressor to the operator gene. So the operator gene is free, and now enzyme RNA polymerase can move from promoter to structural genes via operator gene. So in the last part, we want to see here what is the role of lactose. A few molecules of lactose enter into the cell by an enzyme permease. A small amount of this enzyme is present even when operon is switched off. A few molecules of lactose act as inducer and bind to repressor. This repressor-inducer complex fails to join with the operator gene, which is then turned on. Structural genes produce all enzymes. Thus, lactose acts as an inducer of its own breakdown. When the inducer level falls, the operator is blocked again by repressor. So, structural genes are repressed or inactivated again. This is called negative feedback mechanism. So, whatever the diagram I am showing here, so that is in the form of column. Now, I want to show here the similar type of diagram. It is there from textbook. So now see this is the textbook diagram. Now you compare this diagram and whichever the diagram, whether the previous diagram it is simpler or this diagram it is simpler. So you make it out and you try to use at the time of examination. Thus we completed here the first point, that is called as the regulation of gene expression. Our aim is to study the next part, that is called as the genomics and the human genome project. So that's why whatever this is there in front of you, that notes which are there related to that is regulation of gene here. So this is the next part we want to study, that is called as the genomics. The term genome, introduced by H. Winkler in 1920, is the total genetic constitution of an organism. Alternatively, it is a complete copy of genetic information, that is DNA, or one complete set of chromosomes, monoploid or haploid, of an organism. You are knowing the meaning of haploid. The term genomics, term coined by T.H. Roderick in 1986, is the study of genomes through analysis, sequencing, and mapping of genes along with the study of their functions. The sequencing of yeast, Drosophila, and mouse genome was done in order to facilitate comparative studies between humans and other organisms, commonly used for genetic studies in laboratory. Several additional genomes are now either actively being sequenced or strongly considered for sequencing. These include several microbes, bee, tomato, and other crops. Genomic study may be classified into two types. Number one, structural genomics. It involves mapping, sequencing, and analysis of genome. Number two, that is functional genomics. It deals with the study of functions of all gene sequences and their expression in organisms. So that's why we want to see here, that is the application of genomics here. Structural and functional genomics is used for different purposes in the improvement of crop plant, human health, and livestock. The knowledge and understanding acquired from the genomics research can be applied in a number of different sectors, including medicine, biotechnology, and social sciences. It helps in treatment of genetic disorders through gene therapy. Genomics is used in agriculture to develop transgenic crops having more desirable characters. Genetic markers developed in genomics have applications in forensic analysis. Genomics can lead to introduce new gene in microbes to produce enzymes, therapeutic proteins, and even biofuels. Hence, from this, one new project was established in America, that was Human Genome Project, which is briefly also called as HGP. The Human Genome Project was initiated in 1990 under the International Administration of the Human Genome Organization, which is also called as HUGO. This project was coordinated by the United States Department of Energy and National Institute of Health. International partners in the United Kingdom, France, Germany, Japan, India, and China. The Human Genome Project formally began in 1990 and was completed in 2003. The Human Genome Project is a multinational research project to determine the genomic structure of humans. The main aims of projects are, so we want to see here the main aims of this Human Genome Project. I kept in front of you. Number one, mapping the entire human genome at the level of nucleotide sequences. Number two, to store the information collected from the project in database. To develop tools and techniques for analysis of the data. Number four, transfer of the related technologies to the private sector such as industries. Number five, taking care of the legal, ethical, and social issues which may arise from project. HGP was closely associated with the rapid development of a new area in biology called bioinformatics. The work of Human Genome Project has allowed researchers to begin to understand the blueprint in building and constructing the human genome. As researchers learn more about the functions of genes and proteins, this knowledge will have a major impact in the fields like medicine, biotechnology, and the life sciences. Therefore, HGP is very important. Human Genome Project was provide a complete and accurate sequence of 3 billion DNA has pairs that make up the human genome and to find out estimated number of human genes. Now about 33,000 genes have been estimated to be present in humans. The project was also aimed to sequence the genomes of several other organisms such as bacteria, for example, E. coli, Rhabditis elegans, a free-living, non-pathogenic nematode. Saccharomyces cerevisiae, you are knowing already, it is yeast. Drosophila, it is fruit fly. Plants like rice and Arabidopsis. Mus musculus, that is mouse, etc. Complete genome sequences of these model organisms will be useful for comparative studies that will allow researchers to study gene functions in these organisms. So the table I want to show here, that is comparative genome sizes of humans and other models organisms with the help of the proper chart. Here the chart, there it is in front of you. So now go through this chart. Find out whatever the human beings they are, Homo sapiens scientifically named, that 46 number of chromosomes are there, where estimated gene 33,000 and estimated size, that is base pairs 3 billion. Similar to that, the genes which are there in mouse, in fruit fly, in the plant Arabidopsis, Thaliana, roundworm, yeast, bacteria, it is given. Okay, thus we completed with this, that is called as the HGP. Now our aim is to study the next part, that is called as the DNA fingerprinting. So let's move to that of the next point, that is called as the DNA fingerprinting. So I kept the diagram in front of you. So with the help of this diagram and with the help of introductory point, I want to explain here what is this DNA fingerprinting. Genes present on chromosomes are responsible for determining characters of the organism as well as for inheritance of characters. Due to recombination of paternal and maternal genes, we differ from our parents. Differences also arise due to frequent mutations that occur during gamete formation, that is cell division. Due to all these factors, every individual has its unique genetic makeup, which may be called as fingerprint. The technique developed to identify a person with the help of DNA restriction analysis is known as DNA profiling or DNA fingerprinting. The technique of fingerprinting was first given by British geneticist Dr. Alex Jeffreys in 1984. DNA fingerprinting technique is based on identification of nucleotide sequence present in this wonder molecule. About 99.9% of nucleotide sequence in all persons is same. Only some short sequences of nucleotides differ from person to person. In the population, every person shows unusual sequences of 20 to 100 base pairs, which are repeated several times. They are termed as variable number of tandem repeats. Those are also called as VNTRS. The length of the region having VNTRS is different in each individual and hence is the key factor in DNA profiling. Steps involved in DNA fingerprinting are shown in the diagram, and also I want to explain simultaneously. Please refer the diagram. The diagram I kept in front of you. So the steps involved: number 1, isolation of DNA. The DNA must be recovered from the cells or tissues of the body from the host. Only small amount of tissue like blood, hair roots, skin, etc. are required. Number 2, restriction digestion. The isolated DNA is treated with the restriction enzymes. The restriction enzymes cut the DNA into small fragments having variable lengths. This phenomenon is called restriction fragment length polymorphism, that is RFLP. Number 3, gel electrophoresis. The DNA samples are loaded for agarose gel electrophoresis under the electric influence. The DNA fragments which are negatively charged move to the positive pole. The movement of these fragments depends on the length of the fragments. This results in formation of bands. Double-stranded DNA splits into single-stranded DNA by alkali treatment. Southern blotting. The separated DNA fragments are transferred to a nylon membrane or a nitrocellulose filter paper by placing it over the gel and soaking them with the filter paper overnight. Number 5, selection of DNA probe. A known sequence of single-stranded DNA is prepared. It is called DNA probe. DNA probe is obtained from organisms or prepared by complementary DNA preparation method. The DNA probe is labeled with the radioactive isotopes. Number 6, hybridization. Probe DNA is added to the nitrocellulose filter paper containing host DNA. The single-stranded DNA probe pairs with the complementary base sequence of the host DNA strand. As a result, DNA-DNA hybrids are formed on the nitrocellulose filter paper. Remaining single-stranded DNA probe fragments are washed off. Number 7, photography. The nitrocellulose filter paper is photographed on an X-ray film by autoradiography. The film is analyzed to determine the presence of hybrid DNA. Thus, we have completed the methodology of DNA fingerprinting. Now, what are the applications of DNA fingerprinting? Number 1, in forensic science, DNA fingerprinting is used to solve problems of rape and some complicated murder cases. Number 2, DNA fingerprinting is used to find out the biological father or mother or both of the child in case of disputed parentage. And number 3, DNA fingerprinting is used in pedigree analysis in cats, dogs, horses and humans. Thus, we completed also the applications of DNA fingerprinting. Thus, students, we have completed the lesson which we started this lesson with that of the historical background. In addition to that, we completed the brief account of revision of this one that is called as the DNA. In addition to that, we completed replication of DNA. In addition to that, we completed how the longer DNA it is there, it is kept in the smaller one, that is packaging of the DNA in case of prokaryotic type of cell, in case of eukaryotic type of cell. In addition to that, we have completed the next part that is called the protein synthesis, where the transcription mechanism it is there, we completed translation mechanism, we completed. Meanwhile, in addition to that, we completed about the what about the genetic code, what are the characteristic feature of genetic code, how the gene expression it is there, that is called the how translation mechanism it is there, that we completed. In addition to that, we completed here that is called as the how the regulation of gene it is there, what is the meaning of what you called as human genome project, and in the last part we completed about the DNA fingerprinting. That's why we come to the conclusion of the lesson. Hence, students, we completed the lesson. Thing is that what are the difficulties in studying of this lesson? Some of the important points you have to note down here. See, the various processes are very important. Say number one, it is there, that is how the replication process it is there. The steps involved in the replication process that you have to prepare properly. Not only that, in the transcription process, in translation process also, what are the what you call as the steps which are there. See, in the transcription, the transcriptional unit is very important. Many of the students, they not understand what is the meaning of transcriptional unit. So that's why you are supposed to prepare properly. I already given the diagram. Okay, prepare that diagram, isn't it, properly, and then you can understand properly what is the meaning of transcriptional unit. In addition to that, the various steps involved in the translation process also. So that also you have to prepare properly. See, these all three processes are there, which may be asked for a long answer type question. So I know that the question is now only for 4 marks, but don't think that for 4 marks entire thing will be asked, that transcription will be asked, or the translation, or the what you call as protein analysis it is there. So the part of that only asked, or some definitions are asked, or the diagram will be given, that part you have to describe properly, that will be asked, some definitions will be asked, isn't it? So like this, the 4-mark question will be completed.